This submitted material is a recording plan for a short audio piece exploring how sex hormones like testosterone and estrogen rewire the human brain, specifically targeting behavior, memory, and evolution. As I understand it from the notes, the argument here is that human personality is shaped by an invasion of powerful chemicals, which they introduce with a 1-minute hook. Expanding the discussion to include the brain's localized hormone production provides a more accurate and startling revelation for the audience.
Yeah, because is there a risk that focusing solely on peripheral chemicals might overlook the profound implications of local internal neural synthesis?
Yes, absolutely. That is the exact trap this draft falls into. The weakness here is that the current plan treats hormones purely as peripheral chemicals invading the brain from the outside.
Right, it completely misses the crucial scientific reality that the brain actively synthesizes its own sex hormones.
Exactly. So to elevate this piece, the suggestion is to shift the narrative hook to highlight the brain as an active endocrine organ.
I like that, shifting it from passive reception to active construction.
Yeah, specifically focusing on how hippocampal neurons synthesize hormones like estradiol de novo from cholesterol.
Because the brain isn't just, sitting there waiting for testosterone or estrogen to arrive via the bloodstream from the gonads.
Right.
Neural cells express all the enzymes required for the synthesis of these steroids right there in the tissue.
Which is such a better visual for an audio piece. You want the listener to visualize the mechanics. So for some concrete examples, instead of just saying chemicals travel to the brain, the writer could describe how neuronal aromatase produces estrogen locally.
And you could explain that this is regulated by calcium transients and gonadotropin-releasing hormone, GnRH.
Right, and let's pause on that terminology for a second.
Yeah.
If a writer just drops terms like neuronal aromatase or calcium transients in the first 2 minutes, the listener is going to completely tune out.
Fair point. We don't want to replace a simple hook with a dense textbook reading.
Exactly. To make this accessible, they can explain aromatase as a localized chemical converter, like an enzyme that takes raw materials and converts them into estrogen right inside the brain cell.
Oh, that's good. And those calcium transients are essentially just the electrical sparking of the brain's internal wiring.
That is a much better way to frame it. You're talking about a local manufacturing plant, not just an import business.
And diving into the specific examples of how that local manufacturing works really elevates the scientific rigor.
It does.
For instance, the release of GnRH from specific neurons in the hippocampus is thought to regulate this local synthesis in a sex-dependent manner. This mechanism contributes directly to the sexual differentiation of hippocampal neurons during the perinatal period.
And it has immediate effects on the physical structure of the brain. This local synthesis controls synaptic transmission and spine density. It acts as an internal homeostatic mechanism.
Right, because local neuron-derived estrogen is essential for the normal expression of long-term potentiation.
Which is essentially the molecular foundation of memory.
Exactly. In both male and female brains. But what's really remarkable is how highly specific this operation is.
How so?
Well, while local estradiol appears to be essential for maintaining synaptic connectivity in the female hippocampus, dihydrotestosterone appears to be crucial for the exact same functions in the male hippocampus.
Wow. Which paints a picture of a highly responsive, self-regulating system. Instead of the brain being a victim of circulating chemicals, the brain is pulling the levers itself.
Yeah, shifting the protagonist of the story from the hormone to the brain itself.
It gives the listener a completely different appreciation for their own neural architecture. Now, we should explicitly state that focusing on neuronal aromatase or localized estrogen production, these are just a few out of all possible ways to reframe the opening hook to be more original.
Right. The overarching goal is just to ensure the brain isn't portrayed as a passive sponge.
Exactly. Let's look closely at the section discussing the core narration, where the plan allocates a brief 3 to 5 minutes to explain hormones, behavior, memory, and evolution. Grounding the broad concepts of memory and behavior in the specific physiological differences between male and female brains makes the scientific argument significantly more robust.
Yeah, I was immediately struck by that time frame. 3 to 5 minutes of audio is roughly 500 to 700 words.
It's tiny.
The weakness here is that allotting only 3 to 5 minutes to sweep across hormones, behavior, memory, and evolution risks severely diluting the complex, sex-specific mechanisms at play, particularly regarding hippocampal inhibitory neurons.
It's an impossible structural burden. You cannot cover the entirety of human evolutionary neurobiology in 500 words without sounding like a rushed Wikipedia summary.
So what's the fix?
The suggestion is to narrow the narration's focus, specifically looking at the distinct organizational versus activational effects of testosterone and estrogen on specific brain circuits, such as CA1 parvalbumin or PV inhibitory neurons.
Building on that point, what if we looked at it from the perspective of how those specific neural circuits actually develop differently based on sex?
Yes, that is exactly how you structure this section.
But wait, for a general audience audio piece? Are we sure we want to plunge them into CA1 parvalbumin inhibitory neurons? That sounds like we are falling right back into the textbook trap we just tried to avoid.
It sounds dense, yeah, but it's all about how the writer frames it. You don't have to read the scientific name like a robot. You can introduce these PV inhibitory neurons as the brain's internal traffic cops.
Okay, traffic cops. I like that.
They dictate the flow of electrical traffic, which is what allows a memory to actually form. If you make this specific cell the main character, you can tell the story of evolution and behavior through its lifespan.
So, if the script zooms in on how these specific traffic cop neurons act as mediators of sexual differentiation in the hippocampus, the narrative becomes incredibly grounded.
Exactly. You could use concrete examples to structure this section by contrasting how estrogen regulates synaptic inhibition and memory in adult females, while it does not have the same effect in males.
Right.
And to illustrate this, the writer can reference how neonatal testosterone alters the hippocampus, establishing lifelong sex differences in perineuronal nets and memory.
Perineuronal nets, PNNs, that is a brilliant detail to include here.
It's so visual.
It really is. If the writer's looking for strong visual imagery for an audio piece, PNNs are perfect. They are essentially extracellular matrix structures. Think of them like molecular chainmail or a tight physiological corset.
Molecular chainmail. That's fantastic imagery.
They wrap around certain neurons, particularly these parvalbumin inhibitory neurons, and they restrict plasticity to lock in critical periods of development.
Exactly.
It physically restricts the brain from changing once it has reached a certain developmental stage.
What is crucial for the writer to emphasize here is how sex steroid hormone concentrations affect that chainmail across different brain areas. Manipulating sex steroids before puberty actively alters PNNs in the CA1 region in adult female mice.
The organizational effects are profound. Neonatal testosterone administration actually alters prepubertal female mouse hippocampus-dependent memory, the function of those inhibitory neurons, and the estrogenic regulation of adult female perineural nets.
Which clearly demonstrates that the sex differences in brain-derived estrogen regulation are established by the early organizational effects of neonatal gonadal hormones.
That distinction you mentioned earlier between the early organizational effects and the later activational effects, it creates such a natural, compelling narrative arc for the listener.
It really does. It gives the script a concrete timeline. You have the organizational phase, where neonatal testosterone establishes the baseline structure and puts the chainmail in place. Then you have the activational phase later in adulthood, where adult estrogen regulates the daily synaptic inhibition.
It explains the how and why of evolutionary brain differences without relying on vague generalizations.
Right, and bringing it back to the writer's pacing issue, using the CA1 parvalbumin inhibitory neuron as the central character allows the script to address hormones, behavior, memory, and evolution simultaneously without spreading the science too thin.
You can also see this in other animal models, which might be a compelling aside for the script if they have 10 seconds to spare.
Oh, definitely.
For example, in canaries, testosterone concentrations positively affect PNN expression in areas of the forebrain that correlate with sensory motor vocal learning.
That's fascinating.
Yeah, the structural neuroplasticity here, the physical changes to neural circuits, including the growth of new dendritic spines and the regulation of that chainmail scaffolding, is heavily dictated by these hormonal levels.
And we should make it clear that utilizing PV neurons and perineuronal nets are just a few out of all possible ways of solving the pacing and focus problem. They could choose a different circuit, but they must choose a specific microscopic anchor.
Exactly. They just need something concrete.
So a key theme running through this material seems to be the evolutionary and behavioral themes of the submission. Integrating the concept of lifelong temporal neuroplasticity into the core narrative ensures the evolutionary angle feels immediate and highly dynamic.
That makes sense, but could the evidence also support a different conclusion? Like the idea that this rewiring is deeply cyclical and responsive to a lifetime of environmental factors rather than a fixed trajectory?
Yes, and that gets right to the issue. The weakness here is that the current outline implies a somewhat static or purely developmental takeover by hormones.
Right, it reads like a one-and-done childhood development story. You get your hormones, your brain gets wired, and that's who you are for the rest of your life.
Exactly, which completely overlooks the highly dynamic, cyclical nature of hormone influence across the adult lifespan. The evidence points entirely in that dynamic direction, and the script needs to reflect that reality.
So how should they approach it?
The suggestion is to incorporate the mechanics of temporal neuroplasticity, teasing how these chemicals constantly remodel the brain's physical structure in real time in response to hormonal cycles and environmental stressors.
This is where the concept of neuroplasticity becomes incredibly exciting for a listener.
Definitely.
A lot of people hear neuroplasticity and just think about learning a new language or picking up the guitar. They don't realize it means the biological capacity of the brain to continuously alter its actual anatomical structure.
Right. Functional neuroplasticity occurs rapidly, affecting attention, memory, and perception throughout adulthood.
So what are some concrete examples they could use?
To weave this real-time remodeling into the script, the writer could discuss how dendritic spines actually change density in sync with the estrous cycle.
That is a perfect illustration. The estrous cycle's impact on dendritic spine density shows that the physical architecture of the brain is literally expanding and contracting in rhythm with these hormones.
Expanding and contracting in rhythm.
It is not static, it breathes.
That's exactly the type of phrasing the audio piece needs. And when you introduce environmental factors into that rhythm, like stress or physical exercise, the narrative gains a layer of complexity that directly connects to the listener's daily life.
The interplay between lifestyle, environment, and hormones is a rich vein for the script to tap into. If the brain is expanding and contracting, what happens when we introduce chronic stress?
Well, early life stress can disrupt the normal development of those perineural nets and PV neurons during critical periods. The estrous cycle actually modulates the effects of early life adversity on avoidance behavior through progesterone signaling.
And on the flip side of stress, you have exercise.
Yes, highlighting physical exercise is a phenomenal way to show how environmental factors intersect with hormone levels to physically alter brain-derived neurotrophic factor, or BDNF expression over a lifetime.
We should definitely clarify BDNF for the writer's scripting purposes. It is often referred to informally as miracle grow for the brain.
Miracle grow, yeah.
BDNF supports the growth, differentiation, and survival of new neurons. It is essential in central synaptic plasticity. What is truly remarkable and highly relevant to the script is that the cognitive advantages of exercise and the subsequent BDNF release are heavily influenced by gender-specific hormonal levels.
The mechanics of how exercise achieves this are fascinating, and they tie perfectly into our earlier point about the body's internal manufacturing. When skeletal muscle contracts during exercise, the muscle itself acts as an endocrine organ.
Wow.
It releases signaling molecules called myokines into the bloodstream. One of these myokines, called irisin, crosses the blood-brain barrier and directly upregulates the expression of that miracle-grow BDNF within the hippocampus.
That completely bridges the gap between the body and the brain. And this process intersects directly with the sex hormones the script is centered on.
Exactly. Women may exhibit improved memory and emotional management after engaging in specific training due to the unique interplay between exercise-induced BDNF and their specific hormonal regulation, like estrogen variations during the menstrual cycle, or even later during menopause.
It shows that the listener has agency. In older adults, regular physical activity has been linked to increased hippocampal volume and improved spatial memory, directly countering the age-related decline in both neurotrophic factors and sex steroid hormones.
It isn't just a story about chemicals invading and controlling you. Conversely, chronic stress and the sustained release of high levels of cortisol can result in the disruption of the feedback loop that regulates the hypothalamic pituitary adrenal axis.
Right.
Excess cortisol literally causes damage to the morphology of dendritic spines and the synaptic integrity of hippocampal neurons. By highlighting how sex hormones and neuroplasticity act as a buffer against, or a casualty of this environmental stress, the evolutionary angle becomes immediately relevant. The listener realizes their daily run or their stressful commute is actively interacting with their sex hormones to physically build or break down their brain.
You can see how this cyclical, responsive view of the brain creates a far more compelling audio experience. You have molecular chainmail being synthesized and degraded.
Yeah.
You have dendritic spines blooming and pruning in sync with reproductive cycles. You have muscle contraction sending irisin to the hippocampus to boost BDNF. It elevates the brain from a static computer hard drive to a living, breathing ecosystem.
That's a beautiful way to encapsulate it. And we've thrown out a lot of different avenues here, from muscle contractions to estrous cycles to cortisol damage. We must explicitly state that these are just a few out of all possible ways to demonstrate how dynamic this rewiring truly is.
The writer can pick whichever environmental stressor or cycle best fits their creative vision.
Exactly. To quickly recap the main points of our critique today. First, we recommend reframing the brain as an active endocrine organ that synthesizes its own sex hormones locally to strengthen the opening hook, rather than treating it like a passive sponge.
Second, we suggest solving the severe pacing and focus issues by narrowing the narrative down to the sex-specific organizational and activational effects on a microscopic anchor, like PV inhibitory neurons and perineuronal nets.
And third, we advise highlighting the dynamic, cyclical nature of temporal neuroplasticity to bring the evolutionary and behavioral themes into the listener's present-day reality.
We summarize these actionable suggestions for the listener to implement as shifting the narrative from peripheral invasion to local synthesis, anchoring broad behavioral concepts in highly specific neural circuits, and introducing the real-time physical remodeling triggered by hormonal cycles, exercise, and stress.
We directly invite the listener to submit their revised work back in again for more feedback.